All right. I think, Professor, we're good to go.
All right, so I'll
Um, I'll introduce you to the audience and then we can start. Hello, everyone. This is the IEEE TEMS graduate chapter here at UT Austin. And we are very proud and it's our pleasure to welcome Professor David Soloveichik to our faculty talk series where he'll share his personal and academic journey with us all. To give a quick introduction about Professor Soloveichik, he's the assistant professor at the Department of Electrical and Computer Engineering here at UT Austin. Prior to joining Texas ECE, Professor Soloveichik was a fellow at the Center for Systems and Synthetic Biology at the University of California, San Francisco. He received his undergraduate and master's degree from Harvard University in computer science and completed his PhD in Computation and Neural Systems at Caltech, where his dissertation was awarded the Milton and Francis Clauser Doctoral Prize for the best doctoral thesis. His scientific area of interest is molecular programming, the engineering of complex molecular systems for synthetic biology, nanotechnology, and bioengineering. He's also interested in the underlying theoretical connections between distributed computing and molecular information processing. Dr. Soloveichik was also the recipient of the famous Feynman Prize in Nanotechnology in Theory category from the Foresight Institute in 2012, and the Tulip Award for the International Society for Nanoscale Science, Computation, and Engineering in 2014. And so it's our utmost pleasure to welcome Professor Soloveichik to this talk series. Professor, the virtual floor is yours.
thank you so much for the wonderful introduction. That was, thank you for having me. It's a pleasure. I want to make this interactive, so feel free to put any questions on chat or whichever way, raise your hand or whichever way Zoom allows. So I thought the way to do this is to start with a little bit overview of the research that's happening in my lab, and then to sort of move more broadly about my academic career and sort of just to have a nice chat with you guys about any kinds of questions that you might have, and I'll try my best to give advice if I can. Yeah, just to start out with, just to overview the research that's happening in our lab. I actually have a pre-recorded presentation, so it's a sort of well-oiled that I had from before. So I thought, why not use the Zoom to our advantage here and just play that. So I'm gonna play a pre-recorded presentation, but feel free to interrupt if you have any questions, and I will just pause it and answer it. So. My name is David Soloveichik, and I wanted to talk to you today about some of the work that is happening in my lab. Now, I'm a professor in the Electrical and Computer Engineering department here at UT Austin, but our work is highly interdisciplinary, and I work with students from biochemistry and other programs. Okay, so what is the big picture? I guess the field that I represent is sometimes called molecular programming. So what is molecular programming? Well, the big picture is that we're interested in programming molecules to carry out complex tasks. And to describe it, it sort of helps to contrast with synthetic biology. So both molecular programming and synthetic biology are interested in building complex biochemical systems capable of lifelike complexity and behavior. But synthetic biology takes sort of these very sophisticated, evolved molecular machines and tweaks them, right? As molecular programmers, we try to build functionality bottom up. This means that we have to use very simple parts that are very well understood. So I like to say that, sort of protein enzymes and all these wonderful biological machines are, in some sense, alien technology, in the sense that we can't build them from scratch. Okay, so to give you the kind of idea of the kinds of projects that are happening in my lab, I wanted to share with you 3 projects going from the most sort of experimental to the most theoretical and abstract. Okay, the first project is about building a DNA-only chemical oscillator. So as molecular programmers, I said before, we wanted to start with these Lego-like simple blocks with well-understood parts, right? And the place where we start is with DNA, because DNA has this well-understood Watson-Crick base pairing. Now, we're not going to care about the 3D helical structure of DNA, so we'll represent it abstractly like so. Now, the basic programmable reaction that we work with in my lab is called strand displacement. Okay, suppose you have this two-stranded complex where the red strand is a partial complement of the green strand. What happens when you add the blue strand, which is the full complement of the green strand? Well, it displaces the red strand, the partial complement. So cartoon-wise, we can visualize the whole process like this. Okay, and this is a DNA-only reactions. We don't need any proteins, no enzymes are required, just salty water. Here's a video from Microsoft Research illustrating this kind of strand displacement process. Okay, now the interesting thing about strand displacement is that you can create strand displacement cascades like this. So here, the blue strand initiates this cascade, displacing the red strand, which can then displace the purple strand, which can then go on and displace the brown strand, which can be seen as the output of the whole network. Now, to give you a taste of how even more complex behavior is possible, here's an example of how these two blue strands can cooperate to displace the red strand, right? Each blue strand by themselves is not enough, but these two blue strands together can displace the red strand. And then the red strand again goes through the cascade, resulting in the release of the brown strand. So we can abstractly think about this as sort of an abstract chemical out of them. Well, we thought, wouldn't it be fun to build a chemical oscillator? So what is a chemical oscillator? It's a chemical system that exhibits some kind of periodic behavior over time. Right? Now, of course, in a closed chemical system, you won't be able to oscillate forever, but there are systems that can oscillate for a very long time. It turns out that 3 coupled autocatalytic reactions like this form a chemical oscillator. So all we have to do is to design the strand displacement system which has the behavior like this. So for example, what we want is a strand A and a strand B to start a cascade together, which ends in the release of strands B and B again. Now, putting fluorophores and quenchers in appropriate places, we can use the spectrofluorimeter to record and plot the color change, and then we can infer that the oscillations occurred as we programmed them to. The big picture is that we're beginning to think about chemical behavior as something that can be programmed. We can imagine that synthetic chemical oscillators like ours could one day be used in synthetic biology or completely artificial cells to ensure that certain things happen before others. I now want to mention another application of the DNA strand displacement reaction that we are currently working on, one for DNA storage. Now, you may have heard of DNA storage before. This is an idea that several companies are working on. It's very promising. And the basic idea is that we want to use DNA instead of magnetic media or hard drives and tape drives to store digital electronic information. And of course, the main advantage of DNA is in the storage density, right? It's about 1 exabyte per cubic millimeter, which is about 6 orders of magnitude more than magnetic storage media, for example. Now, the disadvantage of DNA storage is it's inefficient in terms of time and cost, right? And in particular, we can't do any kind of in-memory computation, right? So if you have data stored in DNA form and you want to do some kind of computation on it, information processing, run kind of statistical analysis, something like that, we have to extract information from DNA using sequencing technology, compute electronically, and then resynthesize the new information in the DNA strands to put back into the system. So this is something that we want to address using DNA strand displacement reactions to do in-memory computation on the information stored in the DNA itself. And in particular, the idea is that the information is stored in the identity of the strands that are attached to a long single-stranded template. So this sort of shows the idealized scheme where the information, whether it's a 0 or a 1, is stored whether there's one strand or two strands, two separate strands attached in the corresponding location on the template. And this kind of information representation allows us to use strand displacement reactions to manipulate the information that is being stored in the DNA in a form of computation. So what that looks like is that you add particular instruction strands that then perform strand displacement reactions based on what the data is actually stored. And this computation happens in parallel. All of the registers, as we call them, each storing different data, is manipulated by the same instructions in memory. And what we get is, although the individual instruction steps of strand displacement are relatively slow, sort of in the order of the minutes, we get the benefit of massive parallelism by having multiple registers in the same test tube. So this is a very promising idea of combining basically strand displacement based computation with DNA storage technology to get a high storage density, as well combining with computational capability. Okay, it's a final example of a project that's going on in my group, and this is a theory project. All right, so motivated by being able to program in arbitrary reactions like A plus B goes to C using strand displacement cascades, we note that we can think of the stoichiometric transformation of reactants to products as performing a form of computation. So in particular, C stops getting produced once A or B runs out, which means that computationally speaking, we're computing the minimum of the initial amounts of A and B, right? The amount of C that's eventually produced is the minimum of the initial amounts of A and B. We can look at other examples of such computation by stoichiometry. So for instance, X goes to 2Y, computes multiplication by 2. As we saw before, x1 plus x2 goes to y, so it computes the minimum. We can compute the sum by converting both x1 and x2 to y independently, right? So then the amount of y we get at the end is going to be the sum of the initial amounts of x1 and x2. And we can compose such things into more complicated systems. So for instance, you can think about this offline, that this system computes the maximum of x1 and x2 in the same way. Right, so we can ask, what is the computational power of such computation by stoichiometry, right? And in a sort of surprising connection, we figured out that we can exactly characterize it in terms of something called ReLU neural network. So if you're coming from a machine learning community, these are sort of very popular kind of neural networks that are used everywhere these days. And it turns out that you can show that this computation by stoichiometry sort of has exactly the same computational power in a very, very tight connection to these ReLU neural networks. So this sort of suggests another way that we can use things like strand displacement cascades to do computation in chemistry. And as far as we know, this is not something that biology does. But hey, we don't build airplanes to flap their wings the same way as birds do. So if we want to embed a microcontroller into a cell, we don't quite have to do it in the same way as a cell naturally does. So just to conclude, I think it's important to keep in mind that there are really only three kinds of computing hardware that's prevalent on Earth, right? There's the electronic computers, there are the brains, computation happening there, and then there's computation happening in every single cell of every living organism on Earth within the complex chemical regulatory networks. So a lot of what we do in the lab is we think about this last one as a form of computation, and we ask the kind of questions, how can we create chemical reactions that do information processing, and how can we understand how that information processing can in principle happen in chemistry. Thank you for listening, and I'll take any questions that you guys might have. All right. So thank you for listening. So do you guys have any questions I'll take any questions that you guys might have.
You can feel free to unmute yourself and ask or type your questions in the chat box. Meanwhile, Professor, I have a question. So, in terms of the whole field of DNA computing, um, what do you think is like the holy grail, or what is like your dream? Um,
Yeah, so I think DNA computing is sort of, in some sense, maybe slightly misleading term, because it originated really with Len Adelman's paper in the 90s, which was all about doing sort of NP-complete computation in polynomial time, quote unquote, using sort of massive parallelism of nucleic acids, so DNA. And that's sort of not really, I mean, it's a cool idea, but practically it doesn't really pan out. I mean, sort of not surprisingly, it sort of just trades space for time or time for space, that, you're going to need an exponentially large test tube to solve, complex and complete problems. So sort of that sort of the origins maybe, well, that sort of trajectory of really solving computationally hard problems using DNA computing, I think is really not an active area. But what is sort of a few things. One is, in the context of, if you want to reprogram life in a sense, right? I mean, that's what synthetic biology is about, is to make, life-like things, or to sort of to interface with life, right? Again, like we can't put an electronic microcontroller into the cell, but we can, put a chemical microcontroller into the cell that somehow reads the kind of chemical inputs and outputs that the cell already has. I mean, it already has a chemical microcontroller that we want to interface with it and potentially, change its behavior. and so forth. So the goal is not to sort of to outcompete electronics in terms of do computation faster, but to put computation into context where electronics can't go. Now, there's this project that I talked about in terms of the DNA, combining DNA storage and computing. So for the DNA storage aspect of it, that's actually, I think, a very sort of practically interesting, and companies are sort of jumping on that. It is, 6 orders of magnitude more compact kind of information storage than optical or magnetic media that we have. So doing computation in that context is not, again, it's sort of not really to outcompete with electronics in terms of speed, but it's like, okay, we have sort of this other reason to store information in that way. And then, we want to sort of avoid this loop to having to read it out, which is a very slow process, into electronic form, and then compute electronic, and then re-synthesize the new nucleic acid. So here it's sort of all done in chemistry.
That's interesting.
There's a question, I think. I'm sorry?
Yeah, I think Dimitri has a question for you. Shall I read it, or do you want to? Maybe I can read it. So Dimitri says, "Thank you. Seems like the DNA computing still happens in a tube. Can you envision this going to a more structurally organized computer, maybe as complex and bulky as old lamp computers?"
I guess maybe I'm not quite. I guess one way to interpret this question is, yeah. So if I'm understanding this right, like it's a well-mixed sort of test tube, as opposed to like a cell, for example, which has a lot of compartments, and it's sort of it's not just a well-mixed bag of chemicals. It has a lot of different isolated compartments, and then there's a lot of the machinery, there's different channels that kind of allows things to move back and forth. And yeah, I mean, this is an important goal that people are sort of working toward. It's sort of easier to just have a well-mixed test tube. There's all kinds of theory work, for instance, how do you think about sort of isolating things in different parts? Alan Turing, the Turing machines, was also one of the first people to think about chemistry happening in space in terms of, you may have heard of reaction diffusion systems and things like that, that sort of, explain some biological patterns and things like that. So if I understood that question, yeah, but it's sort of harder to do. So in my lab, we still mostly have just well-mixed, the building the compartments is a bit harder. Although, the people who do like synthetic life and things like that, have some very neat ideas for kind of automatically sort of putting things in compartments.
Mm-hmm. I think following on Dimitri's question, what I was thinking was, is it like right now where the field is, we have the hardware already. We need kind of new ideas in terms of algorithmic thinking, finding new kinds of algorithms, or is there like a whole class of unknown organisms with new kinds of machinery inside them working that we haven't tapped yet?
Yeah, so I actually, I think this is maybe a bit of a controversial question, but sort of my answer would be that this is one field where sort of the theory is ahead of the practice. So another area is, for example, is quantum computing, right? So theory there is also ahead of the practice, right? So people have all these ways, all these interesting ways to build very interesting things, but yet we can't actually build them as well in the lab. So at least from kind of a personal perspective, our ideas definitely often outstrip the sort of the things that we can actually build in the lab. In terms of sort of whether there are some other organisms and things like that. I mean, I even want to open up to more, beyond organisms on Earth. Like, there is some space. Okay, there is some space of organisms that exist on Earth, right? Then there's this potentially larger space of alien organisms. That so we're not surprised by any kind of alien organisms that come. But then there's this even larger space of like everything that's possible in principle. And I actually sort of like to think about that space, right? It's sort of some space of chemical interactions that are capable of doing information processing, but you might not actually, there might not be organisms that actually do that, but it's more like what is possible, right? And again, sort of when we construct the chemical, these microcontrollers to put into a cell, we don't have to stick to what actually exists. What kind of interface is used to store and extract data from DNA? Yes, so that's a very good question. For people sort of outside this field, it's a little bit surprising how, how okay, so there is a lot of technology that went into to reading and writing DNA. Okay, so and it's somewhat surprising. So you may not know. Okay, so this is a fun exercise to show. You can go to a website, IDT DNA is one of these companies. For example, let's see if I can just do a live demonstration here. This is always not gonna work when you do this live. It's probably gonna reject my password or whatnot. Okay, okay, here we go. Okay, so you can go to this website, let's say, right? And you can go to DNA oligos, and you can just, single-stranded DNA shipped to dry resistor suspended to your specifications. You can just order now. And, here you just type the sequence that you want to have, ATT, GET, A, any sequence you want. It has some restrictions in terms of the length. If you want really long things, that gets harder. So for DNA storage applications, you may want to not have things too long. So they check for things like, are you trying to create a pathogen or something like that? So there's some, Department of Defense thing that tries to do these checks. But if not, you can basically just type this in and then add to order, and item name is required, and ships it. It's $10. It's actually, we have an additional discount on this, so it's actually less. And you get, trillions of copies of this strand. So this is now sort of a very high throughput technology that exists. It's sort of the way these things are made is somewhat analogous to inkjet printing. So this is, by the way, the COVID vaccine sort of starts out in this way uh, as well. Of course, then it's amplified in E. coli and things like that. But if you want to create some new sequence, this is what you do. On the other side of the coin, if you want to read it out, again, there's some very, very high throughput ways where you can millions of sort of parallel strands read out at the same time with these really high throughput third or fourth generation technologies. From so these things have both reading and writing have exponential curves in terms of cost and speed and all these kind of things. There's Moore's like law for that also.
That looks interesting. That you can order your sequence of choice.
Yeah. So in there's so yeah again on the reading side, like for example in our lab we have this little device called the nanopore. It's an Oxford. Maybe I can show it. Minion device. Let's see if I can pull it up. Yeah. Yeah, here we go. This is sort of the next generation of these things, is that you basically have this tiny little thing you just connect it to the laptop, put a sample there containing arbitrary DNA that you have, and it basically passes these strands through a special protein that, as the strand passes through, it changes the conductance of the current, sort of depending on what is the exact base that passes through. So you can tell A, T, C, and G apart, and there are thousands of these pores that get passed through at the same time. So you can there's this massively parallel way that you can read things out. So it's technologies like this, for example, that allow you to search for pathogens in the field, like if you're sort of looking, or things like, I don't know if you guys remember when they uncovered the Osama bin Laden a while back, and they immediately did a DNA test on him to tell if that was him. So we don't they don't say exactly what they used, but it's things like this that you can that are now becoming much more common and cheap.
So I think there was like another question by Neil Otpala. Is there an electronic aspect to it as well? I think what you showed now maybe explains that.
Yeah.
So I think we're like above the halfway mark here. So Professor, do you want to maybe, if there are no
Yeah.
questions from the audience um, regarding this half of the talk, maybe we can continue with your academic journey. How you wanna? Yeah.
Okay. Yeah. Again, feel free to stop me or ask any questions. This part of the talk is not well planned. I don't. I have not sort of talked about this before in this kind of a context. So let's see. I just wanted to share. Oops. Sorry, minor technical difficulties. Oh, yeah. Okay. So just a few details about me. So yeah, so that's how you pronounce my names. Soloveichik. It means a nightingale in Russian and in other Slavic languages. So it's a diminutive form of a nightingale. So it's a small nightingale. So I was born in Kiev, Ukraine. Well, at that time wasn't, it was the USSR. And my family moved to the United States in 1990. And we settled in LA, in Los Angeles. And for college, I went to Harvard, majoring in computer science. I think I am probably one of those people who underthinks things. Like I sort of never quite had my career path planned out. I sort of never probably spent enough time thinking about what exactly I'm gonna do, and I always was just sort of did what was interesting to me at the time. I'm not sure I would recommend that, but I'm just sort of saying the way things worked out for me. So for instance, when I entered college, I didn't know what the major would be. I didn't even know if I would be pre-med or not. I sort of was actually for a little bit, but then decided not. I didn't know if I would be interested in physics, math, or biology. I was really good at physics in high school. I decided that I wouldn't sort of take the freshman physics courses in college, and that was a big mistake in the sense that I was then not ever able to catch up on physics. So that sort of eliminated that as a possibility. But I didn't really know what math was, but then I was exposed to it, and in particular in the discrete math for computer science courses. I fell in love with it. In particular, I have to say that I really fell in love with some of the mind-twisting things that I don't know if you guys are familiar with the halting problem, or the more kind of philosophy of math stuff, Gödel's theorem, which is really plays into the computer science aspect of it. But I did always sort of have this biology interest as well. So in high school, I did research over the in some biology labs at UCLA, and I never kind of thought that I could combine the two. So for my PhD, I thought I would be working on the brain, and that's sort of another interest of mine, is to understand how the brain works. although I never pursued, it's one of these things, you just say, that's interesting, right? And well, okay, I guess I got my PhD in that area, although I never sort of, I don't claim to be an expert in that. So that was my goal, but I somehow ended up with the wrong crowd. So at Caltech, instead of pursuing this, what I intended to pursue, I ended up with the wrong crowd. So I ended up with my PhD advisor, Eric Winfrey, who was doing this kind of molecular programming stuff. And I really stayed in this lab because of the environment and the people there was just amazing. This was sort of the first, these people were sort of interested in everything. We could talk about everything. It was just this broad set of interests, and people, brought their, again, interest in, I guess sort of there's a common core of interest in the math, but that could be applied to all kinds of areas. And there were biologists and chemists and computer scientists who kind of all were able to collaborate, which is sort of a very surprising thing, usually. And so I was sort of very surprised that, I can sort of use my wet lab. I learned how to pipette in high school, I guess. And so here is like, I can do computer science by pipetting things in the lab. So that was very cool and sort of surprising to me. So I did my PhD basically in that area, and in the area of molecular programming with my PhD advisor. And then for postdoc, I also sort of didn't quite know what to do. I guess I knew that I wanted to be in the academia, but I didn't quite know sort of how I wanted to draw the lines between being more toward biology or sort of more away from biology, sort of, what space to study in terms of things that actually exist in biological systems, or a larger space of all that could possibly exist. And my wife also had to do a medical residency in the Bay Area at Stanford. So I got a postdoc at UCSF, which is a medical school, if you're not familiar. So I spent a number of years there kind of talking to biology people, sort of these very heavily biology people. And at that point, I probably sort of realized that that's probably not something that I want to do. I want to sort of think more broadly about the space of everything that's possible, as opposed to just concentrate. It's very interesting. So one sort of piece of advice I would have is I think people sort of underappreciate just how different fields are. So things like biology and computer science and things like that. It just in terms of not just sort of, it's in terms of the sort of the basic axioms that people have about what is interesting, right? So there is no, we just take for granted that some things are interesting, that if you go across fields, you can't justify it because it's an axiom, So for example, biology people, just take as an axiom that it's interesting, to figure out how some regulatory pathway in our bodies work, right? they might, when they give a talk, they might sort of say, oh, it might lead to some medications in the future or something like that. But that's not really why they do it. I mean, of course, some are, but if you're, that's somehow fundamentally interesting to understand some, biological regulatory pathway, right? And other fields have the same kind of sort of axiom. So this is one kind of example of trying to communicate across fields. So at that time, I was also trying to prove things. So I love to prove things, and so I was thinking about some kind of information processing that chemistry sort of can or can't do and I was this was a collaborative work. We were proved something was sort of impossible. That sort of chemistry can't compute something in this way, right? And so I was very excited, and I was sort of telling to some biology friends that I had there, sort of explaining what this was all about. I was very excited. And then the biology friend, it's like, nods. And so I think that they understand what I'm talking about. And then they're like, yeah, in biology, we don't publish negative results, right? So, okay, so first of all, that leads to a complete kind of misunderstanding of what this is, right? Sort of prove that something is impossible is not a negative result in the same way that, an experiment did not work out, right? But it just, that bridge is just very hard to cross, right? Why, how do you explain to a biologist that you proved some math result that, so that chemistry can't do thing X, right? So yeah, so this is sort of, I think, again, people maybe I love interdisciplinary work. This is, I'm not trying to say to sort of not to reach out and talk to people across the field, but just be, you have to, in fact, it's the opposite. You have to be a lot more open to trying to understand how the people across the aisle think, right? And that probably is the same thing, sort of more generally. We live in our silos, whether it's the academic field, profession, or political, inclination, and we don't sort of try to understand what the other side, or sort of what are the underlying axioms that they have, and that sort of makes talking to them across the aisle much harder. So yeah, I can sort of continue talking. I don't know if people have any questions.
So I don't see any questions on the chat box.
All right, so advice. Okay, I don't know. I guess some people are looking for advice here. Maybe I don't know if you should ask me, but one thing that I would say is that again, another thing that I think people underappreciate is just how many different paths there are to get to the same sort of end goal, right? So we tend to think about, sort of, you want to achieve X and you take all the usual steps to get there. So for example, in academia, the goal is to become a professor, and you sort of have to follow the standard way, whatever that is, to get there. Get the bachelor's degree, get the master's or not, get the PhD, apply to jobs and things like that. So I guess the point I've seen there's a lot of different paths to get there, and people go into the industry, then come back. One of the smartest people I know, Matt Cook, who I did my PhD with, he actually, never got a bachelor's degree at all. He dropped out of college. He went to, then he did some groundbreaking work with Stephen Wolfram. And then he, was hired. I mean, was accepted into the Caltech PhD program without ever getting a bachelor's. Now he is a faculty member at ETH, which is a top school in Europe. And so people do all kinds of things um, just because sort of something happens to you, or sort of you take a particular um, you know junction in your path. uh, that doesn't mean that sort of path is cut off. And the other aspect of that, I guess there's just a few minutes left. Again, like, I don't just want to say my things. So if you have any question, feel free. But while if anybody is thinking what to ask, I'll just fill it with some more advice, I guess. The other thing is, chaos theory. Again, I'm not saying anything that you guys aren't aware of yourselves, but it may be worthwhile repeating this, emphasizing it, right? That it's very tempting to overthink things because we have this illusion of control over our lives and our paths, right? So if you try to decide what college to go to among options and sort of try to choose the best option, try to decide, should I go to grad school A or grad school B? Should I go into the industry or go into grad school? And you just don't have enough information to make that choice. You're and there is no way to get that information. You see, the randomness in your future life is going to completely overwhelm any kind of predictive power that you might have at that point. So I don't know if this is a optimistic or a pessimistic state of affairs. All right, it's sort of pessimistic because you have a lot less control over the future than you think you do. It's optimistic because at least I find myself stressing out over choices a lot, and I do, but this is sort of one line of thought that I sort of think when I'm sort of trying not to stress out over choices that you make, because the chaos theory, the butterfly that flaps its wings somewhere, is going to have much more effect, right? It's going to, again, like this choice of which school to go to, something like that, it's going to be completely overwhelmed by the particular people you happen to meet, right? You might find the love of your life there or not. I mean, this is a very, very, very specific thing that you just cannot predict ahead of time.
And to put in an unconventional computing paradigm, I don't think it's reversible computation in any manner.
Yeah, very good. Because there are many ways to get to the same place. And,
Yeah.
yeah, it's hard not to sort of plan. I mean, you have to. You can't just give it up. But I guess sort of the point I'm trying to make, and I'm also not the best at this, but the point I'm trying to make is that not, sort of try to go back in time and say, I made the wrong decision here, or something like that, and not sort of over stress about these things. And in the end, sort of try to enjoy. Okay, what can we hope to do is try to enjoy the kind of things that we do, and enjoy the day-to-day life and the research. I mean, again, I probably am coming off quite unlike what I actually am. Actually, in life, I'm quite a stressed out person, in the sense that I stress out over every choice that I make, despite, but I sort of, this is my way to try to stress out less.
I think, yeah, Professor, you gave a very broad overview on in terms of advice, I think. So maybe I think you answered most of the questions that would have gotten through the cracks. So questions usually in the talks are around how between choosing the field of study after undergrad, uh, or have to navigate through postdoc years after grad school if one is like interested in changing fields or something like that. So I think you gave a very pretty broad overview on all those aspects. Um, so I think I see no questions in the chat box. So it seems like they're all satisfied with the advice that you gave. Um, so we are at the 7 p.m. mark. So if no one has any questions, maybe we can end the talk right here. Thank you, Professor Soloveichik. It was our pleasure to it was our pleasure to host you for this talk series, and we look forward to seeing you again in an offline setting when the whole pandemic boils
Yeah,
down.
seeing you guys actually in person and eating the food there. That's the key.
Yeah. Thank you, Professor Soloveichik, and we'll sign off now until our next talk. Thank you. Have a nice